Welcome back to Scientific Study Spotlight. I'm your host, Dylan, and today we're going to be talking a little bit about cold immersion therapy, but more specifically, we're going to dive into the realm of cold shock proteins. So you've probably heard in recent talks and videos a lot about cold plunges and cold therapy and the benefits it can have for things like recovery after a workout or the anti-inflammatory effects it has, which is all great. But I really want to focus more today on the actual proteins that are responsible for those benefits, so cold shock proteins. So let's dive right into a little bit of background here. Cold shock proteins, also known as CSPs, are a group of specialized proteins found in a wide range of organisms, anywhere from plants to bacteria to even humans. And they've gotten a lot of attention around them due to their ability to protect cells during exposure to cold stress. So what do they do exactly? So basically, when an organism is undergoing extreme cold temperatures and is exposed to these extremely cold temperatures, it faces a number of challenges, including the potential to denature proteins or even the disruption of the cell membrane, which can be catastrophic to the cells. So what cold shock proteins do is they step in to safeguard the cellular integrity and they prevent damage from happening. Now, one of the key functions of cold shock proteins is RNA stabilization. So the protein will bind to a single strand of RNA and that'll prevent it from forming secondary structures that could potentially inhibit the cellular processes that it would normally undergo. So by stabilizing RNA, cold shock proteins can ensure that the essential genetic information can still be accessible and function even in these extremely cold environments. So in addition to RNA stabilization, cold shock proteins will play a crucial role in gene regulation as well. They can interact with DNA and influence the transcription of specific genes. This allows organisms to adapt their cellular machinery to cope with the extreme cold and maximize the chances of survival. Now, another fascinating aspect of these cold shock proteins is their ability to protect cellular membranes. So in extreme cold, the lipid bilayer that will form around the cellular membrane can become rigid and lose its fluidity, which is really bad because it prevents molecules from being transported in and out of the cell. So the cold shock protein will bind to the membrane and prevent it from solidifying, maintaining its flexibility and structural integrity, allowing molecules to move freely in and out even in extremely cold temperatures. So now that we have a little bit of background on cold shock proteins, let's dive into today's paper titled Cold Shock Proteins from Cellular Mechanisms to Pathophysiology and Disease. All right, let's break this paper down. So the authors of this paper were looking at different disease pathways and looking at how YB1 was contributing in that sort of regulation pathway of these different diseases. So the first thing the author points out is that cold shock proteins are highly conserved across organisms. You see them in bacteria, humans, plants, really everything except for yeast. So the authors of this paper really looked at human-specific cold shock proteins. So the protein family is called Y-box protein family, and in this particular paper, they really looked at protein called Y-box binding protein 1, or YB1, which is often referred to throughout the paper. So like I said earlier, these proteins are highly conserved, and for humans, they're really important in early development, and then as we grow into a full-grown human being, they're important then as well. In early development, they actually saw that in mice lacking YB1, they didn't make it through the embryonic stage and they died. So it's really important for that early development, and then also really important as we get older. It's going to help in a lot of different pathways, as we're going to see very shortly. This paper has a really good figure describing the amplification feedback loop that occurs when YB1 is stimulated. So we're going to go through this together. So in order for YB1 to become active, there needs to be an extracellular signal or an extracellular stimulus. This could be cold exposure, stress, They have some growth factors included in here too, inflammation, things of that nature. Once that stimulus is received by the cell, it's going to induce YB1 secretion. So YB1 is going to bind to a membrane-associated receptor on the cell, which is going to then induce a signaling cascade throughout the cell, which is going to result in kinase activation. So the kinases are basically just enzymes that are going to catalyze other reactions in the cell. So once the kinases are activated, YB1 is going to undergo a translocation into the nucleus of the cell. Once it arrives in the nucleus, it's going to start transcription of target genes for certain cold shock proteins in response to this cellular stress. The activated cells are then going to secrete this YB1 as well as other cold shock proteins, and it's going to act in either an autocrine way, which is saying that the cell that released the protein, it's going to bind back to a protein receptor on that cell and induce signaling cascades, or it's going to act in a paracrine way where it's going to affect cells surrounding that cell that secreted the YB1. Once the YB1 is secreted, it's then going to recruit immune cells to go fight that inflammation from that original extracellular stimuli. So once the YB1 is secreted, it's going to go take immune cells, go fight the inflammation or whatever the stressor was. So a really cool picture here just to kind of get an idea of that original pathway that YB1 is undergoing, and really it kind of goes back to all those videos we see on cold therapy and things like that, cold plunges, on how exactly it's fighting inflammation once it gets that cold response stimulus. The last part of this paper I want to talk about is the rationale for cold shock protein targeting for certain diseases, mainly cancer. So in this paper, they list 4 examples. I'm only going to go over 2. But essentially, the 2 I want to look at are the one where YB1 is regulating what's called IL-2 production. So IL-2 production is needed in order to activate T cells, which are responsible for, which is a type of immune cell, which is responsible for killing cancer cells. So IL-2 production is needed in order to stimulate what's called CD28, which then in turn will do its signaling cascade and then activate T cells. So YB1 is responsible for mediating that IL-2 production. The other interaction that they look at is YB1 and P53. So P53 is a really common mutation in a lot of cancers that occurs. So P53 is a tumor suppression protein that is responsible for basically regulating the cell cycle. So if something goes wrong in the cell cycle, P53 pathway is typically activated, and then typically it'll either try and fix itself or it's going to induce apoptosis. So what YB1 does is it doesn't actually affect its regulation of that pathway. So if something still goes wrong, it's going to say, okay, we need to fix this or try and fix this. But what YB1 actually affects is P53's ability to induce apoptosis or cell death. And that can be really detrimental. So even though it can still regulate and tell the cell, hey, you need to fix this, it still may not be able to kill the cell if need be, if they can't fix the problem. So just 2 really interesting areas of target that they looked at. So really, for example, 2, you would want to increase YB1, you want to upregulate YB1. That way you can increase IL-2 production, increase T cell production to get rid of the cancer cells. And then for the P53 example, you'd want to downregulate your YB1. That way it can still induce apoptosis if necessary. So just really two interesting pathways that they looked at in this particular paper. So really interesting paper. I highly encourage you guys to read the whole thing. And the last little thing I want to touch on is the developing topics in the cold shock protein field. So just a really quick couple of examples that they list. So they're looking at pro-inflammatory factors that can play a role, and they're looking at negative feedback loops that help relieve chronic inflammation with these cold shock proteins. And then the other one, which was really interesting, is that cold shock proteins can help regulate formation of protein aggregates. And these are really common in neurodegenerative diseases, so things like Alzheimer's, dementia, things of that nature. So they're looking at how they can use YB1 to sort of regulate that pathway. So really interesting stuff. So thanks for watching, guys. Really appreciate it. If you liked what you saw, hit the thumbs up and don't forget to subscribe. If you have any topics you want us to take a look at next, be sure to put it in the comments below and we'll be sure to take a look at it. Thanks for watching, guys. We'll see you next time.